Hi, I'd like to thank CTSI for this opportunity to present my work. My name is Christina McConney, and I'm a PhD student in the lab of Dr. Snyder-Capion in the microbiology department. Today, the title of my presentation is Longitudinal Measurement of Antiviral and Autoimmune Antibodies for 2 years after SARS-CoV-2 infection reveals uniquely durable vaccination responses and partial waning of self-reactive IgG. So here's my poster of my work, and I'll be expanding upon these figures in further detail in the coming slides. In this study, we were measuring the changing levels of autoantibodies and antiviral antibodies for as long as 2 years after COVID-19. So abnormally high levels of autoantibodies are often present in people hospitalized for COVID-19, and these antibodies can bind a wide range of self-antigens that can actually dampen the immune response to virus. Further, there are reports of people with mild COVID-19 also possessing high levels of autoantibody. Also, there's further reports of new onset autoimmune diseases and related conditions after SARS-CoV-2 infection, and autoimmune responses might actually be driving past or long COVID. What we still don't know is to what extent these antibodies are newly induced from SARS-CoV-2 infection versus pre-existing in these individuals. And of the antibodies that are induced by this infection, it's still unclear if the expression pattern wanes after acute illness, and if so, when it happens. Another big question about COVID-19 at this time is the role of booster vaccinations. So first vaccinations given in 2 doses were roughly 95% effective at preventing infection. And we learned by the summer of 21 that a booster was needed to restore protection at that high level after Delta variant emerged. However, this protection waned after the first few months, and then Omicron emerged, leading to 90% of the US population becoming infected with SARS-CoV-2 in the past 9 months. And now we have a booster available that is matched to current variants in circulation. However, some question how effective this booster will be at preventing infections and thereby transmission, and for how long. Also, just this month, a study was published in the New England Journal of Medicine showing that healthcare workers who were first vaccinated and then had a breakthrough infection maintained neutralizing antibodies for longer than the uninfected counterparts. So given this, in this study, we were looking to see if infection occurs prior to vaccination, will there be less waning of the vaccine-induced SARS-CoV-2 spike antibodies? And if this is the case, we could predict that the new booster, given on the backdrop of infection-induced immunity, could have longer lasting benefits than we are currently anticipating, provided that protection covers shifts in variants. So we have 2 main questions in this study. The first being, do autoantibody levels wane over time after SARS-CoV-2 infection? We have previous work where we measured autoantibody trajectories for 6 months after infection, and that manuscript is available on medRxiv. And in this work, we are following some of the same cohort for longer periods of time, up to 2 years after initial symptom onset. Our second question asks, if one is first infected with SARS-CoV-2 and then vaccinated, will those antibodies be long-lived, meaning will they remain at high levels for a year or more? So our COVID-19 study participants are from 2 groups, those who are hospitalized with an N of 40 and those who are outpatient, N of 33. In our original study, we measured autoantibodies from plasma that was collected at multiple time points during the beginning at the first week of symptom onset and for an additional 6 months. From this larger cohort, we obtained more samples for some of the subjects from later time points after they had received COVID-19 vaccination and who were followed for a total average of 20 months post-symptom onset. So just to note here, all of these subjects had COVID in the first year of the pandemic, so they were all infected with the ancestral strain of SARS-CoV-2 and had not received vaccinations. To measure autoantibodies, we used a commercially available kit from Millipore Sigma that detects IgG antibodies that bind 17 self-antigens found in people with autoimmune diseases of connective tissues and associated conditions. We then used the ELISA, which is a sensitive protocol developed in-house to measure IgG and IgA levels reactive to a panel of viruses and associated antigens. So here's data from our medRxiv manuscript showing how individual autoantibody levels changed in SARS-CoV-2 infected subjects during those first 6 months. And we have 5 autoantibody expression patterns defined here, with blue representing positive at all time points, gray representing negative at all time points, red indicating a shift from negative to positive, the inverse shown in purple from positive to negative, and then green shown as changing repeatedly at all time points. Here's a heat map of all of the outpatients, so all 33 subjects, with each patient ID shown vertically and the autoantigens detected shown horizontally. And each colored box here represents the determined autoantibody pattern per antigen in each subject. So for outpatients, the majority of autoantibody hits were positive for all time points, and the induced and sustained autoantibodies shown here in red were found in only 6 subjects, so that's 18%. However, among the inpatient group, more subjects had this induction and sustained expression pattern over the 6 months, totaling 53%. So here, as I mentioned, part of my project is to determine if these elevated autoantibody levels remained high after 6 months. And at the top are just visual representations of the autoantibody expression pattern definitions that I'll be referencing. So below, I show autoantibody expression trajectories found in all 7 subjects over an average time span of 20 months after symptom onset. And looking across these 7 subjects, what's noticeable is the predominance of purple and green patterns among the subjects that were not present in the first 6-month study. So overall, there's a waning of autoantibody levels between 6 months and 2 years after infection. And when we quantify this, we find that of the 25 autoantibodies marked as positive at all time points in those first 6 months, 8 of them waned below what was scored as a positive hit. So that's 32%. Whereas in those that were initially marked as induced and sustained in the first 6 months, only 3 of them stayed high at these later time points, with 78% waning to a negative score. We also found that trajectories of autoantibodies vary within the same subject. For example, here we see an instance of an autoantibody that was induced and sustained even 25 months post-symptom onset, another that was then induced and then waned at later time points, a third that showed an increase initially and then maintenance of a positive autoantibody hit in the later time points. And finally, we see one that was initially marked as positive at all time points, then dramatically fell after a year. What I want to point out here is the ramping up of all 4 autoantibodies during these first 6 months or so after infection, and then the very different trajectories that each of these took. However, overall, they all seem to be falling just at very different rates, and some much more slowly than others. And so now I'm going to switch gears and talk about our antiviral antibody results. And here we have representative data from what we collected so far from subjects with multiple time points after their first COVID-19 vaccine. So from a currently processed N of 3, which includes 1 hospitalized and 2 outpatients, all 3 showed marked increase in anti-spike IgG after vaccination that was sustained for at least 11 months. More importantly, there is little to no sign of waning of these vaccine-induced antibodies. So this pattern of antibody expression over time is different than that of nucleocapsid, shown here in green, which will be induced by infection but not vaccination. These anti-nucleocapsid antibodies are highest at the first time points of infection for all 3 subjects, and then wane before leveling off, with the exception of OP2, whose anti-nucleocapsid IgG antibodies actually fell to undetectable levels, but then this patient was reinfected, causing a spike in that antibody. Also, when we look at antibody levels to other viruses from the same subjects at all time points, we see very steady levels during this long time period. So in conclusion, most autoantibodies waned after more than 1 year after infection, and SARS-CoV-2 infection followed by vaccination was shown to lead to durable anti-spike antibody responses for at least 11 months post-vaccination. The study was limited in that there are no parallel longitudinal samples of individuals infected with other viruses, disallowing us to say how unique these results are to SARS-CoV-2, and there are no samples available pre-infection from the subject, so we can't confirm pre-existing antibodies. Our next step include increasing subject numbers to determine if links between magnitudes of IgG and A antiviral antibodies and autoantibody trajectories exist, as well as phenotyping B cells from blood draws to track spike-specific B cell memory populations and determine shift frequency with help from Shift Ply of the Reagan Institute, as well as measuring cytokines and other analytes for composite signature readouts. So there's lots of people to thank. I want to start with thanking my advisor, Jen Capione, and everyone in the Capione lab, as well as Vicky Sidleski for help with the experiments. Also, it's important to thank Nahid Bedelia, who had the initial idea to follow autoantibody levels from these subjects. And I'd like to thank members of the BMC for support with this project, the members of Mass CPR, Autoimmunity Inflammation Working Group. And I'd also like to thank the study participants, Zach and Liv Duffy, for help with COVID-19 sample selection and our funding sources.